Hello and welcome to National 5 Biology, Unit 1, KA6, Respiration. We're still on Unit 1, Cell Biology, and this is our final topic of Unit 1, Respiration. Once again, here's the SQA course specification or mandatory knowledge section for this topic. This is all the bits of knowledge and content that you could be tested on in a test or exam at this level for this topic. As you can see, this is a bigger topic than the previous one, and again we're going to split this up into smaller chunks and do some questions after each part so that you can be sure you're ready to move on. So our learning intention today is to learn about respiration, and hopefully by the end of this lesson you'll be able to describe the stages of aerobic respiration and fermentation in animal, plant and yeast cells. I also hope you'll be able to state the location of these stages inside the cell, and finally describe how a respirometer is used. The first key thing we need to know is what respiration actually is. One common misconception or mistake people often make is thinking that respiration is breathing. This is not the case. At National 5 level, we describe respiration as a series of enzyme-controlled reactions that is used by all cells to release the chemical energy stored in glucose. So let's break this down further. So in KA2, transport across cell membranes, we learned that glucose is a key molecule required by cells in order for them to survive. We take in glucose through our food, and it is key that we have enough of it so that each of our cells can get it. This is because glucose contains chemical energy, and that's really important. You may have learned about the types of energy before using mnemonics like Professor Shekel, so different types of energy you might know are things like light, potential, or kinetic energy, among others, but you need to know that glucose is stored as has chemical energy inside of it. When cells receive the glucose, they must release this energy using a series of enzyme-controlled reactions so that they can use it. We covered enzymes in Area 4. Remember, enzymes speed up chemical reactions in living cells, so enzymes are involved in and speed up these reactions as part of respiration that break down the glucose inside cells. Because they're enzyme controlled, this also means that respiration can be affected by factors like temperature, which, if you remember, affect the enzyme's optimum conditions and the rate of activity, and we'll come back to this again a bit later. So chemical energy stored within glucose must be released by all cells through a series of enzyme-controlled reactions that we call respiration. So what's the purpose of respiration? Why do cells need to break down this glucose in the first place? Well, the chemical energy released from the breakdown of glucose is used to generate a really important molecule that's called ATP. ATP is a molecule that is used to transfer energy in a cell. So it holds on to energy, and it can be used to we can use that energy later by using that molecule. And the energy that's transferred by ATP can be used for various cellular activities. So activities that take place in the cell that need energy to be able to happen. Now, you don't need to know all of the activities in a cell that require energy from ATP, but these are the 4 you have to know at National 5 level. So ATP is needed first of all by muscle cells for contraction. So if you think about when you move a muscle in your body, your muscle tissue is made up of lots of tiny muscle cells that can contract and relax. They need energy to be able to contract, so the muscle cell contraction is our first cellular activity that needs energy that's provided by ATP. Our second is cell division, and we're going to come back to this in a lot more detail in a whole topic in Unit 2. So cells dividing into 2 daughter cells. Number 3, our third cellular activity that requires energy from ATP is protein synthesis, which we've already covered in Area 3 DNA. So ribosomes basically need energy to be able to make those proteins that we talked about before. And finally, cells need energy to carry out transmission of nerve impulses. So this is where nerve cells pass information through electrical signals or impulses. And again, we're going to come back to this in Unit 2 when we look at the nervous system. So please remember these 4 activities that need the energy from ATP to be able to happen, and to get that ATP, respiration has to happen, so the breakdown of glucose. So there are 2 types of respiration you need to know about: aerobic respiration and fermentation. And the main difference between these 2 types is that aerobic respiration can only occur if oxygen is present and needs oxygen to take place, whereas fermentation can occur in the absence of oxygen. So aerobic respiration and fermentation are the 2 types. Be careful if you're using older textbooks because they might mention anaerobic respiration. We never use that term anymore, just aerobic respiration or fermentation. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions either by saying them out loud or by writing them down. And when you're ready, play the video again and we'll go through the answers. Okay. So for number 1, it's important you mention all of the different parts. So it's asking what is respiration, And we know from earlier it's a series of enzyme-controlled reactions that releases the chemical energy that's stored in glucose. For number 2, the energy released from the breakdown of glucose is used to generate which molecule? We know that's ATP. For number 3, state the 4 examples of cellular activities that require this energy. Muscle contraction, cell division, protein synthesis, and transmission of nerve impulses. You need to know all 4 to be able to recognise them. And for number 4, where is the site of aerobic respiration in a cell? Well, I never told you this one just yet, but if you think back to KA1, the site of aerobic respiration is the mitochondria. I need to know this for the next bit we're moving on to. Okay, so before we move on to the main stages of respiration, there's a very common question I want to mention how to complete. So exams and tests often ask you to explain why the energy requirement of different cells are not the same, or why cells have different numbers of mitochondria. To answer this, remember if a cell needs more energy, so has a higher energy requirement because of its function, the cell will have a higher number of mitochondria because mitochondria are the site of aerobic respiration. So for example, let's imagine a question asks you why sperm cells contain more mitochondria than skin cells. Your answer would be that sperm cells contain more mitochondria than skin cells because mitochondria need more energy to be able to carry out their function. So they need more energy to be able to swim to fertilise the egg. So now we're going to move on to the main part of the respiration topic, which is the stages of respiration. For each stage of respiration, you need to know the substrate, the product or products, the location of that stage, the number of ATP produced, which is also known as the yield, whether oxygen is required or not for that stage to take place, and finally the overall word equation or summary for that stage. This list is a great way to test yourself for this topic, so try and list for each stage these parts and then mark them, and this will get you used to the types of questions you could be asked in an exam. Now, although you learn respiration just through lots of paragraphs and words, you can do that. I find the best way to learn respiration and these key facts for each stage is by visualising it in a diagram just like this one. So I've used this diagram for a few years now for my classes, and I find if you can eventually draw this out from memory, you'll be able to answer almost any other questions on respiration other than the ones I've just covered. To properly revise respiration, I suggest drawing it out from memory on a piece of paper and then checking against the marking scheme and then put in anything you missed. Then a few days later, try the same thing again until you can draw this out completely with no mistakes. In previous years, my pupils have found respiration a really difficult topic, and they've decided that as soon as they were allowed to write an exam on the back blank page for notes, then basically they would write this out. And that would mean that they could turn back to this for any questions on respiration that they came across. So now what we're going to do is we're going to break down this diagram into individual stages, which I'll explain one by one, and then we'll come back and we'll put this all together again at the end. So first we're going to look at stage 1, which comes right at the top of this diagram, as it occurs in all cell types and in both types of respiration, aerobic and fermentation. So no matter which type of cell or which type of respiration you're looking at, glucose is always first broken down into 2 molecules of a molecule called pyruvate. As a result of the glucose being broken down into these 2 pyruvate molecules, 2 ATP are also made, which you can see coming off the side here with the curved arrow. So we can say that stage 1 yields 2 ATP in total. As this happens in both types of respiration, this first stage happens in the cytoplasm, and it doesn't require any oxygen. As if it did in this stage, it would only be able to happen in aerobic respiration and not fermentation. So glucose is broken down into 2 molecules of pyruvate, which also yields 2 ATP. This doesn't need oxygen, it happens in the cytoplasm, and it occurs in every cell type. Now let's try some questions on that. So pause the video here and without looking back, try and answer these questions about stage 1 by answering them out loud or writing them down. And when you're ready, play the video to get the answers. Okay, so for question number 1, it asks what is the yield in ATP in stage 1? And we know that's 2. It also asks, does stage 1 require oxygen? No. It happens whether oxygen is present or not, which is why we find it in both fermentation and aerobic respiration. Number 3, where does the first stage of respiration happen in the cell? It happens in the cytoplasm. Number 4, what is glucose broken down into? It's broken down into 2 molecules of pyruvate. Okay, so stage 1 has now happened, generating 2 molecules of pyruvate out of the original glucose molecule. Now, further breakdown of these pyruvate molecules depends upon 2 different factors. 1, whether or not oxygen is present, and 2, which type of cell respiration is happening within. So first, we're going to look at what happens to the two pyruvate molecules if oxygen was present. So this is aerobic respiration and can happen in all cell types. So we're focusing on this pathway on the left-hand side here of the diagram. So this slide summarises in a diagram and words what happens in stage 2 of aerobic respiration along with a word summary on the bottom left. So aerobic respiration happens in all cell types and can only happen if oxygen is present. That's why it's called aerobic. The two pyruvate molecules that were made in stage 1, so that would be up here, but I've cut it off in the diagram, move into the mitochondria of the cell, which is the site of aerobic respiration as we know from KA1. Once it's in the mitochondria, the two pyruvate molecules are further broken down into carbon dioxide and water, and this breakdown also releases a large number or many ATP. You don't need to know the exact number of ATP generated from aerobic respiration. If you see this number in a textbook, video or notes, they're from the old course, as you no longer need to know it. The final thing you need to know about this stage is the word summary, which you can see on the bottom left. So if you're asked for a word summary equation for aerobic respiration, you would write this. So glucose always goes first, as this is what we started with at the beginning of stage 1. All of the word equations for respiration always start with glucose. That's an easy place to start. We also add oxygen, since this is aerobic respiration, so glucose plus oxygen. And in aerobic respiration, this gives us carbon dioxide plus water plus energy. Energy will be used to generate ATP, but we just write energy in the equation. So the word summary for aerobic respiration in total is glucose plus oxygen, arrow, carbon dioxide plus water plus energy. Really important you do an arrow and not an equal sign as well. So now let's try some quick questions on stage 2 of aerobic respiration. Pause the video here without looking back, try and answer these questions by saying the answer out loud or writing them down, and when you're ready, play the video to get the answers. Okay, does aerobic respiration require oxygen? Yes, it does. It's aerobic, so it needs oxygen to happen. Where does aerobic respiration take place in a cell? Well, we know that basically oxygen is like the key to the mitochondria. Aerobic respiration, site of aerobic respiration is the mitochondria. What is the pyruvate broken down into during aerobic respiration? Carbon dioxide, water, and energy. Number 4, what is the overall equation for aerobic respiration? Glucose plus oxygen, arrow, carbon dioxide plus water plus energy. And finally, for number 5, how many ATP, or what is the yield of ATP, produced during aerobic respiration? We just have to say many, so many ATP. Okay, so now we've looked at what happens after stage 1 if oxygen is present in any cell type, aerobic respiration. Now what we're going to do is we're going to look at 2 possible pathways that could occur when oxygen is not present. So if oxygen is present, it always goes from stage 1 into aerobic, but if oxygen is not present, there's 2 options here. So first we're going to focus on the pathway in the middle, which is fermentation in animal cells. So in the absence of oxygen, after stage 1, where glucose was broken down into 2 pyruvate molecules, fermentation takes place. In animal cells, the pyruvate molecules are converted into a substance that's called lactate, and this second stage doesn't make any ATP. We say the overall yield of ATP from fermentation in animal cells is 2 because we still have the 2 that we created in that first stage from before. So this process of fermentation in animal cells produced only 2 ATP in total, which is much less than the many ATP that we produced in aerobic respiration. This is why cells do not want to do this process if possible. One way to remember lactate is produced in animal cells with no oxygen present is by thinking about what happens when you exercise. So your body is made up of animal cells, and if you exercise a lot, you might sometimes find that your muscles get sore. You may also know that this is because of a buildup of something called lactic acid inside your muscles. Now, lactic acid and lactate are basically the same thing, but you have to say lactate in National 5. The lactic acid or lactate builds up and makes the muscles sore as you're not getting enough oxygen to your cells when you exercise, and this is why your breathing rate increases a lot when you try to get oxygen to your cells so that they can do aerobic respiration instead. This is also why when you finish exercising and your muscles are a bit sore, you kind of pant quite a lot and breathe in a lot to get in loads of oxygen, and eventually your muscles aren't sore anymore because you start to do aerobic respiration instead in your muscle cells, and so the lactate's actually gotten rid of. So because the mitochondria is the only site of aerobic respiration, and pyruvate can only move into the mitochondria if oxygen is present, remember try and think of oxygen as being a key into the mitochondria, well, oxygen isn't present in animal cells during fermentation. So in fermentation like this one, It just takes place in the cytoplasm, just like in stage 1. So the pyruvate stays in the cytoplasm and then is broken down into lactate. Finally, you need to know the word summary for fermentation in animal cells, so down on the bottom left. We still start with glucose, like all types of respiration, but this time there's no oxygen, so we don't add it here. Be careful with that. So glucose instead is broken down, and we show this by an arrow, into lactate and energy. Now let's try some quick questions on stage 2 of fermentation in animal cells. Pause the video here again, try not to look back, answer the questions by either saying them out loud or writing them down, and when you're ready, play the video to get your answers. Okay, so question 1, does fermentation in animal cells require oxygen? No, it doesn't. It's fermentation, so it's not needed. Number 2, where does fermentation take place in an animal cell? It's in the cytoplasm, because remember, we can't get into the mitochondria. Number 3, what is the pyruvate broken down into during fermentation in animal cells? Lactate. Remember, not lactic acid, lactate. Number 4, what is the overall equation for fermentation in animal cells? Glucose, arrow, lactate plus energy. And then finally, how many ATP are produced during fermentation in animal cells overall? Overall, it's 2, but remember, those 2 come from stage 1, not stage 2. So now we're on to our final pathway in respiration. This is the fermentation pathway that occurs when no oxygen is present in plant and yeast cells. So this section that's circled in purple is what we're going to look at now. So plants and yeast cells obviously don't have muscles. So lactate can't be produced in fermentation when oxygen isn't present. Instead, plants and yeast, the pyruvate molecules from stage 1 are broken down into carbon dioxide and ethanol. Now, there's 2 ways to remember this. As humans, we harness this fermentation process in plants and yeast all the time. For example, to make alcohols like beer or wine, we ferment plants. We don't provide them with oxygen, and as a result, they produce ethanol and carbon dioxide. The carbon dioxide diffuses into the air, and we're left with ethanol or alcohol. Please make sure, though, that you don't say alcohol in the exam. It's ethanol. Another example of how we use fermentation is when we make bread. If any of you have ever made bread before, you'll know that you have to use yeast to make the bread rise. Again, when yeast isn't provided with sufficient oxygen, it makes ethanol and carbon dioxide through fermentation. Now, this time the carbon dioxide gets trapped in the dough, and that's what makes the dough rise. You might wonder why bread, though, isn't alcoholic, because it's still producing carbon dioxide and ethanol by the yeast. But the ethanol actually burns off or evaporates at really low temperatures. So the ethanol produced by the yeast in the bread evaporates when it's baked in the oven, and that's why bread isn't alcoholic. But the yeast are still producing ethanol and carbon dioxide. So in fermentation in plants and yeast cells, carbon dioxide and ethanol is made from the pyruvate. The other details are the exact same as fermentation in animal cells. It takes place in the cytoplasm, it happens with no oxygen is present, and it only yields 2 ATP in total, both of which were made back in stage 1. Finally, you need to know the word equation or word summary for fermentation in plants and yeast, and that is glucose, arrow, carbon dioxide and ethanol, and energy. Remember not to put oxygen at the start because this is fermentation. Now let's try some quick questions on stage 2 of fermentation in plants and yeast. Pause the video here without looking back, answer these questions by saying them out loud or writing them down, and when you're ready, play the video to get the answers. The first question, does fermentation in plant or yeast cells require oxygen? Nope. Number 2, where does fermentation take place in a plant cell? Cytoplasm, again, just like it was in an animal cell. Number 3, what is the pyruvate broken down into during fermentation in yeast cells? Ethanol and carbon dioxide, and you can say and energy here too if you want to. Number 4, what is the overall equation for fermentation in plant cells? Glucose, arrow, ethanol plus carbon dioxide plus energy. And finally, number 5, how many ATP are produced during fermentation in yeast cells? That would be 2 ATP overall, both of them coming from stage 1. Okay, so now we've been through all of the stages in more detail individually. Here's what it looks like when they're all put together. Stage 1 is the same in every cell and occurs whether oxygen is present or not. Glucose is broken down to 2 molecules of pyruvate, and this breakdown yields 2 ATP. This happens in the cytoplasm. What happens for stage 2 below this line depends on the type of cell and whether oxygen is present or not. If oxygen is present, all cell types do aerobic respiration within the mitochondria, where they break down 2 molecules of pyruvate into carbon dioxide, water, and many molecules of ATP. If oxygen is not present, however, fermentation happens, but what is produced depends on the cell type. So in animal cells, the pyruvate molecules are broken down into lactate, but in plant and yeast cells, they're broken down into ethanol, carbon dioxide. In both cases, no oxygen is present, the location is still in the cytoplasm, and no ATP is generated in the second stage, but we still have our 2 from the start, so you get 2 ATP overall as the yields for these. Here's also a helpful table that I think summarises the differences between aerobic respiration and fermentation quite well. Really important to know this, so either with the diagram or with the table, because it could be an extended response question. They could ask you, describe the differences between aerobic respiration and fermentation for 4 marks, and you would need to explain some of these in detail. The final thing we need to learn about for fermentation, or for respiration, is a piece of experimental equipment. So National 5 Biology, you need to be familiar with the mandatory equipment or equipment list. Any of these can be asked about in questions in the exam, and you would be expected to know already how they work. They don't have to describe it in the question to you. So in order to measure respiration in living things, a special piece of equipment called a respirometer is used. This allows us to measure the rate of respiration, or how quickly it's happening. Both of these diagrams show respirometers. The one on the left is a simpler one and more like the ones you'll see in past papers or tests. However, I wanted to show you the one on the right too in case this came up. The one on the right also contains a control tube, and we'll come back to this in a second. So respirometers are set up with the following parts. Inside a boiling tube, there is a gauze platform where some form of living thing is sat on top of it. So this could be worms, it could be maggots, woodlouse, any other insects, or even living things you wouldn't necessarily think of. So an example that comes up quite a lot here is germinating peas, and they would be living things too and would sit on this platform. The boiling tube is then sealed with a bung at the top, and this prevents air getting in and out. And the only gap contains a thin glass tube called a capillary tube, which has water or a coloured droplet inside it. Essentially what happens is the living organism will take in oxygen to be able to do aerobic respiration and will give out carbon dioxide. However, the carbon dioxide given out is absorbed by solution A in this diagram. So at the bottom of the boiling tube, a substance called sodium hydroxide is added. And what this substance does is it absorbs any carbon dioxide that the insect produces during respiration. Because the insect is taking in oxygen and the carbon dioxide it gives out isn't replacing it because it's being absorbed by this stuff at the bottom, then it means that this causes a drop in the air pressure, which causes the water droplet of the coloured liquid that's in the tube at the top to move towards the insect. So this air is getting taken into the tube, so this water droplet then moves along. The distance this moves can be measured using a ruler and can be divided by the time that it took for the droplet to move a certain length or for how long you're doing the experiment for, and that will give you the rate of respiration. Now this setup can be used to test various factors, so you could test the type of insect and which one has a higher rate of respiration, or you could look into something like the temperature and how it affects respiration rate. Remember, respiration is enzyme controlled, so we'll have a quicker or a faster rate at some temperatures and we'll have a lower rate at others. Finally, if you were to set up a control for this experiment to prove that the liquid wouldn't move on its own unless respiration was taking place, so it's the insects that are doing respiration or living things that are doing respiration which is causing the liquid to move, you would replace the living thing with either a dead version, which would therefore not respire, or you could replace the living thing with glass beads of the same mass, as glass beads are not living, so won't respire, so hopefully the water droplet wouldn't move, which would prove that it's the respiration of the living thing which is causing the change. And that's what you can see in this more complicated setup on the right here. So on one side, on the left-hand side, is the living thing, and on the other side we have the exact same setup, but this is a control with the glass beads being the only difference. Therefore, we would expect that because the living insect is respiring over here, glass beads don't respire, then this side would have a change in pressure, and the liquid in this tube in the middle would move up towards the left-hand side instead. So that's us finished learning about respiration. I hope you can now describe the stages of respiration along with their locations in the cell, and that you now understand what a respirometer is and can describe how it works. As I mentioned at the start, this is a really difficult topic, so please feel free to go back and watch parts of this video again in the future if you need a refresher. It's not an easy one to get first time. You need to take notes, you need to practice drawing this out over and over and over again. Thanks once again for listening.